All right, the second thing that you need to know, the general overview is we're using glucose. Glucose is that 6-carbon ring that is the breaking down, so the catabolism of carbohydrates, so those polysaccharides into monosaccharides into glucose, and using that to make ATP eventually. But the thing about ATP is generally we need an intermediary. So the electrons aren't transferred directly to ADP, so the diphosphate to make ATP. There are intermediaries used, and these two intermediaries are important that you know what these are. Nicotinamide adenine dinucleotide. So NAD for short. So from here on out, it'll be NAD. And on every thing you see from me that would be testable, you're probably going to see NAD. And then flavin adenine dinucleotide, FAD. They're both forms or thought of as parts of or forms of B vitamins. And they are considered cofactors. They are what will carry these electrons to allow ATP to be made eventually. So those you remember both of those, NAD and FAD. And then they occur, remember these reactions occur together. The results in reduction, so reduction is gain, so oil rig of the coenzymes, those coenzymes are going to be reduced. They are going to gain electrons from the hydrogen ion in this process. It then results in increased potential energy in these two coenzymes or cofactors, and then energy is taken from those which are then, I know they're reduced, but they're considered oxidized substrate from the oxidized substrate glucose in this example. So glucose is the oxidized ones, then the coenzymes are the reduced ones. You really don't have to follow that, but you do need to know that these coenzymes, NAD, FAD, are the electron acceptors. Without those, this whole process would not go on, and without glucose. Glucose not only is not just what we chose, what's chosen here, it is actually the preferred form of fuel for this Krebs cycle slash citric acid cycle slash TCA cycle. Most of the glucose is going to be in the form of monosaccharides. And just to refresh, those are absorbed into the villi of the small intestines, carried to the liver, and then carried to the liver via the hepatic portal, so carried to the liver in the bloodstream, and then taken into the cells with the help of insulin. This is a general formula, not to be particularly memorized yet, or even memorized at any point. But here's the first time we introduced this this concept, this term cellular respiration. So this process of oxidizing glucose to form ATP is known as cellular respiration. Respiration because you actually are going to create carbon dioxide. That carbon dioxide that's created is the carbon dioxide that you breathe out. That's the result of all your metabolic behaviors. You're starting out with glucose, one glucose, outside the membrane of the mitochondria. And if we haven't said this yet, most of this ATP is made within the mitochondria. Mitochondria, remember, we call that the powerhouse in 141. We call it the powerhouse because this is the producer, or where most of your ATP is produced. Glucose is first acted on in the cytoplasm, We call that glycolysis. And then the substrates of that particular reaction then can eventually go into the Krebs cycle or the CTA cycle or the citric cycle, and then be acted upon with regard to having these electrons that then can be passed down what's known as the electron transport chain. We're going to go into each of these pieces one at a time. So the first step is in cellular respiration is to oxidize 1,6-carbon molecule, and what that 6-carbon molecule means is that you have 6 carbons in that ring, and then also remember those carbons are numbered. Eventually there'll be another term to know, and in order to understand that you need to know that we do a 6-phosphorylation. Where that phosphorylation takes place takes place on the number 6 carbon, and that is broken in half. And remember, this would be breaking down, so you're also releasing energy into 2 3-carbon molecules, which we call pyruvic acid. And it happens in the cytoplasm, and we call this process glycolysis. So glycolysis happens in the cytoplasm, and it produces 2 pyruvic acids. One other thing that's very important to remember about this is oxygen is not required. We didn't need to, this is not an aerobic behavior. So this can happen in the absence of oxygen, and so it's a way that you can make ATP, not too much of it, if you have an oxygen debt, due to whatever your oxygen debt may be. I guess the most positive way to have an oxygen debt is because you've exercised too much. And then glucose then is going to, this kind of goes back again, a little bit of a backtrack. I talked about this a little bit already in the last slide, is that you have a concentration gradient. So what can happen is the cell can keep taking glucose in, but it maintains the concentration gradient by actually putting a phosphate group on that 6 carbon. So you're taking the active component off the 6 carbon. Remember, carbon can have 4 bonds. You're breaking one of those bonds, putting a phosphorus molecule, a phosphate molecule on, and that then makes sure that you can bring more glucose in. Another phosphate group is then added to form 1,6-diphosphate. So now you have 2 of them on there, and each of that phosphate group requires 1 ATP of energy in order to be made. So we're spending energy to make this. Hopefully we're going to make some energy. And then on that particular molecule, the one that has 2 added phosphoruses on it, is going to be oxidized to recoup ATP. So it's going to be oxidized. Remember, oxidation is loss. And what you're going to have then is ATP making 2 ADPs are going to be, you're going to add a phosphorus to 2 ADPs, so you're actually going to make 4 ATPs. So the end product of glycolysis is 4 ATPs, but remember you use 2 to make them, so the net gain is 2 there. And the other important thing is this is where you make your cofactor or coenzyme, and that's NAD. You make NAD. Very important, those 2 things. You make some energy, not a whole lot. Can't do much with that. But you make the cofactor NAD. Actually, I should say the cofactor NAD now has a hydrogen ion on it. The second step depends on the availability of enough oxygen. So if you have plenty of oxygen, you're breathing, and you're not working out, then that pyruvic acid can be turned into acetyl-CoA. Acetyl-CoA means you will take another one of those carbons off pyruvic acid, and then it can go into the Krebs cycle. If not, lactic acid is going to be formed, and that lactic acid then is going to have to be removed from your body. If it's plentiful, it's going to go into the Krebs cycle, as I said, and that we're assuming you have plenty of oxygen, so we're going into the Krebs cycle. That's the third step. It becomes a 2-carbon molecule. So 2-carbon, the acetyl-CoA is actually a 2-carbon molecule at that point from the oxidation of the 3-carbon molecule of pyruvic acid. 2 molecules of CO2 are given off, so 2 molecules of carbon dioxide are given off, and then 2 molecules of NAD are produced. So energy is produced and stored as 2 molecules of NADH. Eventually that H is going to allow an electron to go into the electron transport chain, which is where most of the ATP is going to be able to be produced, and that NAD will live again to go through the cycle again to pick up another electron and move it through again. And I had to put a picture over another picture because the one that I used originally wasn't very clear, so that's why it kind of looks like this. But acetyl-CoA goes into the mitochondria. So everything prior to this happened in the cytoplasm. So now the acetyl-CoA is going to go into the mitochondria, where the 2 fragmented acetyl-CoA is dropped off, and I actually said that wrong. The 2-carbon fragment is dropped off, and the CoA is free to diffuse back and pick up another one. So it actually is just a carrier into the mitochondria. The CoA is just a carrier into the mitochondria. Each cycle, so each turn, that's why they call it a cycle, is the 2-carbon acetyl fragment becomes completely oxidized. So going toward the I guess the right of the original equation that we looked at, high oxidation, yielding ATP, FAD, and NAD, FADH2, 2 hydrogens, ions, and NADH. So that would be what you want to have in your head also, is you have complete oxidation of that fragment yielding ATP plus those 2 cofactors, FADH2 and NADH2. NADH, sorry, I keep on saying things wrong, which I'm really apologizing about because it's I don't want to confuse you. So yielding ATP, we don't know how many yet. Eventually we will know, but cofactors FADH2 and NADH. The 4th step then is the electron transport chain, abbreviated as the ETC, but frankly most people call it the electron transport chain. And that is a system for actually pulling off that stored energy that we put onto the FAD and the NAD. The H2 on the FAD and the H on the NAD are considered stored energy. It's composed of special integrated carrier proteins. These are integral membrane proteins embedded in the inner membrane of the mitochondria. And the mitochondria is a organelle that is taken in. It has a double membrane, and one of the reasons why it can function, and we got to stop now and move on to the next one